the Mendel's observations, or why the Mendel's experiments were not published, are not known to the world properly in those days. Okay, there are four main reasons of that. The first one is okay, the communication was not that much effective in those days, because the only communication available in those days is a newspaper. So because of the lack of proper communication, and the second one, the concept of factors of the genes, which are said to be stable and get inherited, okay, from one generation to another generation, this concept of a stable inheritance is not accepted. Why? Because, okay, we are observing a lot of variations in the organisms. We are observing a lot of variations in the organisms. Because of that, okay, this concept of stable inheritance is not acceptable. And next, the usage of mathematical expression in explaining the, okay, probability of genotypes in the organisms is not understood by the biologist at that time. And the last one is okay, the Mendel has not given any physical proof for the factors of the genes, okay, which are involved in controlling the characters. So because of all these four reasons, the Mendel's experiments were not popularized in those days. But later, okay, okay, in the year 1900, okay, in the year 1900, three scientists from three different countries, okay, like a Hugo de Vries, it is given as a de Vries, that is a Hugo, Hugo, Hugo de Vries. And next, Carl Correns, Carl, Carl Correns, and von Teschermark. Okay, that is okay, okay, Hugo de Vries, Carl Correns, and von Teschermark independently rediscovered Mendel's results on the inheritance of character. That means whatever the experiments that were conducted by the Mendel, okay, he has he has published certain journals now. So they have gone all those journals that were given by this John Gregor Mendel, and next by studying that experiments. Okay, these three scientists have reconducted all those experiments, restudied all those experiments. Okay, and finally, they have given the results. Okay, these three scientists, who are those? Hugo de Vries, Carl Correns, and von Teschermark. Independently, they have done the experiments independently. That means they have not worked together separately. Okay, all the three scientists have worked on the Mendel's experiments, okay, to study the inheritance of characters. And also by this time, due to advancement in microscopy that were taking place, scientists were able to carefully observe cell division. So by year 1900, okay, because of the advancement in a microscope, that means we have developed different types of microscope with high resolution. So we are able to study the chromosomes. We are able to observe the chromosomes, and they are able to study the behavior of chromosomes during cell division. They studied the cell division, mitosis as well as meiosis, and observed the behavior of chromosomes during the cell division. So because of the advancement in microscopy, they clearly studied the cell division, okay, and the behavior of the chromosomes during the cell division. This led to the discovery of structures in the nucleus that appear to be double and divide just before each cell division. So when you observe here, okay, that means with the advancement in microscopy, they observed the nucleus and the components of the nucleus, and they found that the components that are present in the nucleus are supposed to be get doubled before cell division and later get divided. Okay, later get divided. Okay, that is these were called chromosomes. Okay, whatever the structures that are present inside the nucleus of the cell, okay, which are supposed to get doubled before cell division and later get divided, and these structures they call it as chromosomes. Okay, that is colored bodies as they were visualized by staining. Colored bodies as they were visualized by staining. And by 1902, the chromosome movement during meiosis had been worked out. So that means by 1900, okay, the Mendel's experiments were rediscovered. Okay, and later in the year 1902, the cell division was studied and the behavior of the chromosomes during the cell division was also studied. So by 1902, the chromosome movement during meiosis has been worked out. Okay, during meiotic cell division was studied. Okay, and next, after observing all this, in the year 1902, okay, two scientists, that is Walter Sutton and Theodore Boveri. Okay, Walter Sutton and Theodore. Okay, Walter Sutton and Theodore Boveri. These two scientists in the year 1902, by observing the movement of chromosomes during the meiosis, okay, noted that the behavior of chromosomes was parallel to the behavior of genes and used chromosome movement to explain Mendel's laws. So what they have, that means just like how chromosomes are present in pairs, in the same manner, the genes which are controlling different characteristics, they are also present in pairs in an organism's cell. Okay, and they are okay, and they used this chromosome movement to explain the okay Mendel's laws. Okay, whatever the laws that were proposed by the Mendel, they were explained with reference to this chromosome movement. Okay, as chromosomes and genes both appears in a pairs in the organism. It was okay, and this was done by the two scientists, Walter Sutton and Theodore Boveri. Okay, that is. And next, recall that you have studied the behavior of chromosomes during mitosis and during meiosis. Okay, that means once if you recall the mitotic cell division and meiotic cell division. Okay, when you observe the mitotic cell division, during mitotic cell division, what happens? Okay, when you observe the cell cycle is divided into four phases: G1 phase, S phase, G2 phase, and M phase. Okay, when you recall the cell cycle, okay, the cell cycle is divided basically into four phases. Okay, that is a G1 phase, S phase, G2 phase, and M phase. G1 phase is the phase during which the cytoplasm content of the cell is increased. Okay, here nothing will happen related to cell division. Just the cell will increases or grows in size, increasing its cytoplasm content. And next, after G1 phase, we'll find the S phase. S phase means a synthetic phase. This is the phase during which the replication of the DNA will occur and the chromatin material is doubled, but chromosomes are not doubled. Okay, chromosomes are not present at this stage. We will find only the chromatin material or genetic material. Only the DNA is doubled. The amount of DNA, that means if the DNA content will become like 4C. But at this stage, we don't find chromosomes. So there is no increase in the number of chromosomes or no change in the number of chromosomes. Okay, and that increase in the genetic material or the, okay, will take place during the S phase. After S phase, we'll find the G2 phase. G2 phase is nothing but the phase during which the cell will prepare for cell division. Okay, whatever the proteins, enzymes, and all other components that are required for cell division are produced during are synthesized during a G2 phase. After G2 phase, now the cell will enter into M phase. M phase means division phase. The cell will undergo division. First, okay, when you observe the M phase is again divided into two phases, karyokinesis and cytokinesis. Karyokinesis means the division of a nucleus. Nucleus means here the chromatin material. Okay, when you observe the karyokinesis is again divided into four phases: prophase, metaphase, anaphase, and telophase. During prophase, what happens? The chromatin material condenses. Okay, the chromatin material starts condensing, and the condensed chromatin material is broken down into small fragments called chromosomes. Okay, and the nuclear membrane, nuclear membrane, nucleolus, and all other cell organelles will disappear. And next, after prophase, the metaphase. During metaphase, the condensation of chromatin material. So the early phase of the early stage of metaphase is the correct stage to study the morphology of chromosomes. That means if you want to study the structure and shape, okay, of chromosomes, the best stage is the early stage of metaphase. Okay, and during metaphase, what happens? Okay, the spindle apparatus is formed by the starting of the metaphase. That means by the end of prophase only, the spindle apparatus is formed. And now what happens means the chromatin, okay, these spindle fibers during metaphase, they will get attached with the kinetochores of the centromeres of the chromosomes, and they are pulled in. Okay, they start pulling. As a result, the all the centromeres of the chromosomes are placed at the center of the cell, called metaphase plate. Okay, with the formation of metaphase plate, the metaphase is completed, and later it enters into the anaphase. During anaphase, the spindle fibers which are attached to the kinetochores of the centromeres, they pull in opposite direction. As a result, the centromere and the chromosome splits, and the chromosomes are separated, and they start moving towards the opposite poles. Okay, depending upon the type of the chromosomes, okay, the chromosomes may attain either V-shaped or L-shaped or a I-shape. Okay, okay, and the movement of the chromosomes towards the opposite poles will be completed by the end of anaphase. And next, after anaphase, the telophase is nothing but it is a quite opposite to prophase. We can say that is the decondensation of chromatin material, formation of nucleus or nuclear membrane, and the formation of cell organelles. All they will be done. And after telophase, it is followed by the cytokine, that is the division of cytoplasm, and finally we'll get two daughter cells, that is prophase. But when it comes to that is about meiosis. But when it comes to meiosis, okay, meiosis is a very complex of all the cell divisions. When you observe here, the meiosis is again divided into meiosis 1 and meiosis 2. Meiosis 1 is again divided into prophase 1, metaphase 1, telophase 1, and anaphase 1. Okay, among all the four phases of meiosis 1, or karyokinesis of meiosis 1. Prophase is a more complex and more prolonged phase, which is again subdivided into 5 phases like leptotene, zygotene, pagotene, diplotene, diakinesis. Okay, leptotene, zygotene, pagotene, diplotene, and diakinesis. Okay, further. Okay, and it is a complex process during which, during leptotene, the chromatin material which is in the form of network, now it starts condensing and it is converted into chromosomes. And next, after leptotene, we'll find zygotene. During zygotene, what happens means the pairing of homologous chromosomes will occur. Okay, this is a pairing of homologous chromosomes. We call it as synapsis, with the formation of a synaptonymal complex among the homologous organs. The synapsis will can occur, and this paired homologous chromosomes now, okay, enters into packaging stage, during which the crossing over will occur and exchange of genetic material, which is mainly, okay, this process is very, very important, which is involved in developing recombinations or new variations in the organisms. And next, after pactine, it is followed by diplotene. During diplotene, what happens? The centromeres split, but the chromosomes are still attached with the region where the crossing over occurs. As a result, the chromosomes appear as an X-shaped structure, which we called as chiasmata. And next, followed by the diakinesis. During diakinesis, the terminalization of this chiasmata will occur. Okay, and during this process also the nuclear membrane disappears, cell organelles disappear, nucleolus disappears, and finally it enters into the metaphase, phase 1. Okay, metaphase, anaphase, telophase, the same almost. And finally, during anaphase, what happens? The homologous pairs separate. The paired chromosomes get separated during anaphase 1. That's why out of the two sets of chromosomes, one set will enter into one cell and another set will enter into another cell. That means the diploid cell is now converted into haploid and followed by telophase and cytokinesis. Telophase 1 and cytokinesis, which results in the formation of two haploid daughter cells. And these two haploid daughter cells that are formed at the end of meiosis 1 now enters into meiosis 2, which is similar to that of mitosis or equational division. And finally, at the end of meiosis, we'll get four daughter cells with a haploid number of chromosomes. That is what the men, okay, this Walter Sutton and Theodore Boveri has studied, and they used this mechanism for explaining this Mendel's laws. So that is, so the important things to remember are that chromosomes as well as genes occur in pairs. Okay, important thing that we have to remember is that, okay, the genes and chromosomes both will occur in pairs. So the two alleles of a gene pair are located on homologous sites on homologous chromosomes. Okay, when you observe here, when you observe a chromosome may consist of many genes. Okay, a chromosome may consist of many genes. Okay, the particular position on the chromosome which represents gene controlling a character, okay, that we call it as a locus. Okay, locus is nothing but the position of gene on the chromosome. Okay, and next when you observe this chromosomes, so that means as we'll find the organisms are diploid, so we'll find two sets of chromosomes. If we take one chromosome, whatever the genes, first for example, if you take first chromosome, whatever the genes that are present on the first chromosome, the same genes or its contrasting alleles are present on the other set of first chromosome. So we'll have two pair now, two chromosomes as pair. So whatever the chromosomes that are present on one chromosome, whatever the genes that are present on one chromosome, the same or similar type of genes are present on the other chromosome also. Okay. So by 9, okay, so that is what we can see here. So the important things to remember are that chromosomes as well as genes occur in pairs. The two alleles of a gene pair are located on homologous sites on homologous chromosomes. Homologous sites means on the same position, on the same locus. Both the genes are present on different chromosomes, but at the same locus, which are called as such genes or such chromosomes which possess similar genes, similar locus, then such chromosomes are called homologous chromosomes. Okay, for example, okay, suppose this is one chromosome. Okay, this is another chromosome. For example, if we take, okay, suppose A gene is present here. Okay, in the same manner, for this also, the A chromosome is present. B, C, D, E, yeah. Okay, like this, different genes are present at different positions on one chromosome. Okay, if you observe the opposite, the other, okay, the paired one, that homologous pair in that one is having A, B, C, D, E, F. Okay, in the same manner, in the other one also, we can observe the similar type at the same position. Okay, at the same position, we can observe the genes. Okay, there is. So here A, here A, here B, here B, here C, here C, or sometimes, okay, it may be, it's contrasting allele may be present, but both are representing only one character only, no. So such, okay, such chromosomes which are having similar genes at the similar locus, then such chromosomes we are calling it as homologous chromosomes, and such homologous chromosomes only will pair. And that, okay, and this pairing of homologous chromosomes and later followed by crossing over, that is the exchange of genetic material, okay, will results in the, okay, that is that separation, okay, during a meiosis, what happened, this homologous pairs only get separated and out of the one, okay, out of the pair, one will enter into one cell and other set, other set will enter into another cell. Like that, how many number of chromosomes are present? For example, if you take human, in human we'll find 23 pairs of chromosomes. So whatever the chromosomes that are present in the one set of 23, the similar chromosomes are present on the other set of 23. Okay, we don't find that much variation. Okay, even if any variation is there, that will be it will form as a contrasting allele, that is the opposite allele, like capital A, small a, okay, capital C, small c, capital E, small e, like this. But they are representing only one characters. That's why we are saying, we are taking into consideration. Okay, that is. And next, they said that the behavior of the chromosomes and the behavior of the genes is similar. Here you can see both chromosomes as well as genes will occur in pairs. Both chromosomes as well as genes occur in a pairs. Okay, we say that. That means, okay, we'll say, we are saying no, whenever I ask what is the chromosome number in a human beings, means we say 23 pairs or 46. Okay, in the same manner, as there are two sets of chromosomes. So whatever the genes that are present on one chromosome, the similar genes are present on the other chromosome also. So in a diploid organism, usually the genes also occur in a pairs. And next, when you observe the separation, segregate at the time of gamete formation such that only one of the each pair is transmitted to a gamete. Okay, that means, when you observe, okay, the chromosomes are separated now. Cell, when it is converting into haploid cell, what happens? The chromosome, okay, whatever the chromosome that are present in the cell, they are forming as pairs, and these pairs, out of these pairs, one set will enter into one cell and another set will enter into another cell. So the segregation at the time of gamete formation such that only one of the each pair is transmitted. So out of the pair, only one is transmitted to the offsprings or to the gametes. Okay, and next, independent pairs segregate independently of each other. Okay, independent. That means if the chromosome has been segregated, whatever the genes that are present associated with that chromosome also get separated. So the independent pairs segregate independent. So then, okay, you can mention as a chromosomes. What A represents means chromosomes. Okay, and when you're coming to the genes, okay, like chromosomes, genes also occur in pairs, but okay, when you observe here also the separation or segregate at the gamete formation and only one of each pair is transmitted to a gamete. So that the transmission is the same. Okay, how it is getting transmitted, that means how it is getting segregated, it is similar to that of chromosomes. But when it comes to segregation, one pair segregates independently of another pair. Okay, one pair segregate independently of the other pair. That means if chromosome, okay, if you take a chromosome, for example, here we can see the chromosome A, B, C, D, E, F is there. Here what happens? Okay, in this chromosome, first A, B, C will be there, and in the next chromosome D, E, F, these two will get combined. So why it is happening means it is because independent pairs will segregate independently. So one pair segregates independent of another pair. And next, during Anaphase 1 of Meiosis 1, the two chromosomes pairs can align at the metaphase plate independently of each other. Okay, during anaphase of meiosis 1, that means that during anaphase 1, the two chromosome pairs can align at the independently. And next, independently of each other. And next, understand this. Compare the chromosomes of 4 different color in the left and right columns. In the left column, orange and green is segregating together, but in the right hand column, the orange chromosome is segregating with the red dinosaur. Okay, red. Okay, with the red chromosomes. Okay, here we can see. Okay, independent pairs segregate independent of each other. Okay, the genes. But when you observe the chromosomes will segregate as a total set. Okay, that means the 46 chromosomes are there. That is 23 pairs. 23 will enter into one gamete, 23 will enter into another gamete. But not in case of gene. Gene is restricted to a small structure. Okay, and next, Walter Sutton and Theodore Boveri argued that the pairing and separation of a pair of chromosomes would lead to the segregation of a pair of factors they carried. So what this Walter Sutton and Theodore Boveri has said, that means when the chromosomes are getting segregated, whatever the genes that are associated with that chromosome, they are also get distributed, and they are and next, Sutton united the knowledge of chromosomal segregation with Mendelian principles and called it as the chromosomal theory of inheritance. So when you observe this Walter Sutton, after understanding the behavior of chromosomes during the cell division, what he came to conclude, that means, okay, the segregation is taking place separately. And he combined this Mendelian principles with that of a chromosomal theory and called it as a chromosomal theory of inheritance. Okay, chromosome. And following this synthesis of ideas, experimental verification of the chromosomal theory of inheritance by Thomas Hunt Morgan and his colleagues led to discovery the basis for reaction. Okay, basis for the variation that sexual reproduction produced. Okay, following the synthesis of ideas, experimental verification. That means whatever the ideas that were given by Walter Sutton and Theodore Boveri. Okay, this Thomas Hunt Morgan has made use of those ideas, and he has given the experimental verification for chromosomal theory of inheritance. Okay. Okay, because of all these experiments, he was considered as the father of modern genetics. Considered as the father of modern genetics. Okay, and next. Okay, with the experiments that were conducted by this Thomas Hunt Morgan, okay, he has discovered that the basis for variation is that nothing but sexual reproduction. So the sexual reproduction, the organisms which are produced as a result of sexual reproduction are the main basis for variation. And next, when you observe the Morgan worked out with the tiny fruit flies, that is Drosophila melanogaster. Okay, what is the organism that the Mendel has selected means? Okay, that the Thomas Hunt Morgan has selected means that is nothing but fruit fly, which is commonly called as fruit fly, and its scientific name is Drosophila melanogaster, which were found very suitable for such studies. Okay, so for his best, okay, for his studies to understand better, he used the organism that is the Drosophila melanogaster. Okay, Thomas Hunt Morgan. Morgan worked with the tiny fruit flies, that is Drosophila melanogaster, which were found very suitable for such conditions or such studies. Okay, because the Drosophila melanogaster is a small organism, it is very easy for handling. So they found, okay, the Drosophila melanogaster is very suitable for his experimental studies and grown on simple synthetic medium in the laboratory. So the eggs can be grown. Okay, this fruit flies will lay eggs, and these eggs can be easily grown in artificial nutrient medium in the laboratory. Simple artificial medium in the laboratory. They complete their life cycle in about 2 weeks. That means they are having very short lifespan. So we can able to observe many generations within a short period of time for studying the inheritance pattern. Okay. And next, okay, they can, okay, to complete their life cycle, usually they take 2 weeks. And a single mating could produce a large number of progeny flies. So once the male unites, okay, once the male unites with the female, or once male the female, okay, a large number of progeny can be produced. Also, there was a clear differentiation of sexes, and we can able to observe clearly which is male, which is a female Drosophila. Okay, that is so differentiation of the sexes, the male and female flies are easily distinguishable. Also, it has many types of hereditary variations that cannot be seen with how power microscopes. Okay, also it has many types of hereditary variation that can be seen with a low power microscope. That means if you want to study any variation, that means if you want to study the generation, the offsprings that are produced, you can easily study by using low power microscope only, by using like hand lens. By using hand lens only, we can observe certain differentiations between male and female Drosophila. That's why, because of all these reasons, okay, this Thomas Hunt Morgan has selected hybridization experiments on a Drosophila melanogaster. So while he was studying about the dihybrid inheritance pattern in Drosophila melanogaster, they have proposed different terms. Okay, they introduced different terms. They explained in the different concepts. Like one out of them, the important ones are linkage and recombination. Linkage and recombination. What is meant by linkage? Linkage means the physical association between the different genes that are present on one chromosome. That are okay. So Morgan carried out several dihybrid crosses in Drosophila to study genes that were sex linked. So that means what the Mendel has, or what this Morgan Thomas Hunt Morgan has done means he selected certain characters that are observed in Drosophila, which are present on only on X chromosome. Because okay, this Drosophila melanogaster is showing the sex determination as XXXY type. Okay, so XX is male, sorry, XX is female and XY is male. So that means the X chromosomes are present in both males as well as females. That's why, okay, she has selected this, sorry, he has selected this characters which are present on X chromosome by the Thomas Hunt Morgan. Okay, Morgan and his group knew that the genes were located on the X chromosome. So that is sex linked. Whatever the characters that they were selected, those are present on X chromosome. The crosses were similar to the dihybrid cross carried out by the Mendel in peas. So the okay, he has undergone the similar way. Okay, that means he has selected homozygous individuals as parents, and he crossed those two parents and obtained the F1 generation. And to obtain the F2 generation, he selfed the F1 generation. That means he undergo the cross between the both individuals of that generation. So that means the crosses that were conducted by Morgan are similar to that of crosses that were conducted by Mendel. Okay, for example, Morgan hybridized yellow body, white-eyed females to brown body, red-eyed males and intercrossed their F1 progeny. He observed that the two genes did not segregate independently of each other, and the F2 ratio deviated very slightly, significantly from the 9:3:3:1 ratio. Okay, that means we observed some different ratios. Drosophila melanogaster when they are conducting the dihybrid cross with the different characters. Okay, Morgan and his group knew that the genes were located on the X chromosome. Morgan and his group knew that the genes were located on the X chromosome and saw quickly that when the two genes in a dihybrid cross were situated on the same chromosome. Okay, Morgan and his group knew that the genes were located on the X chromosome and saw quickly that when the two genes in a dihybrid cross were situated on the same chromosome, the proportion of parental gene combinations were much higher than the non-parental type. So that means when the Morgan has selected certain genes which are present on X chromosome, which are representing the different characters, what he has observed means when the genes are very close to one another, whatever the characters that were selected, there, okay, if these characters, the genes which are representing these characters, if they are very close to one another, he found that, okay, he found a large number of parental combinations, and the new combinations are very less. The recombinants are very less, whereas the parental combinations are more in number. When the genes which are present on one chromosome are very close to one another. When he has selected the genes which are present distantly on the same chromosome, that means if the genes which are present on the same chromosome but they are distantly arranged, in that case, okay, in that case, okay, he found there is a decrease in the parental combination. Okay, the parental, the percentage of parental combinations is less when compared to the recombinants or new combinations. The new combinations percentage is increased when the distance between the genes on one chromosome are long. And this physical association between the chromosomes, okay, between the genes that are present on the chromosomes is described as a linkage. Is described as a linkage. Okay, that means if the genes which are present on one chromosome are very close and close with one another, then we say the linkage is strong. In that case, we'll find more parental combinations and less recombinants. At the same time, if the genes which are present on one chromosome are distantly arranged, then we say the linkage is weak linkage. In that case, the percentage of recombinants is increased and the percentage of the parental combinations is decreased with the first case. Okay, in the first case, more parental combinations and less recombinants. But in the second case, we'll find more recombinants and less parental combinations when compared with the other ones. Okay, that is. And this physical association, we are calling it as linkage. And the new combinations that we are getting in the F2 generation, okay, this new combinations is called as recombinants. And these recombinants are produced as a result of recombination. Okay, that is. And we'll discuss in detail with the experiment, okay, in the next class.
Sir,
Yes, ma'am.